intervention, and those who just have the symptoms, but during the cath lab examination we didn't find an infarct artery, we didn't find a target for intervention. So at first, yes, we send all the patients to the cath lab in order to have more data to decide which are these predictive parameters which could exclude some part of this cohort and consider that the others are more likely to have an MI with an occluded coronary artery.
Thank you so much, Dr. Asimova, for this fantastic talk. Thank you so much.
Thank you too.
Would there be any other questions for Dr. Simova's presentation? If not, I would like to open the floor to present, to invite Dr. Jeremy Owen Go of the Capital Medical Center in Philippines to deliver his talk entitled Beta-3 Adrenoreceptor, Boon or Bane in Heart Failure. Dr. Go?
Hello, am I being seen in the video? The audio is clear? Yeah, so can I share my screen? Yeah, so everyone can see my slides. Okay, so thank you for inviting me as a speaker to today's event, the Scholars Webinar on Cardiology. My name is Dr. Jeremy Go. I am a third-year internal medicine resident in Capital Medical Center, Philippines. And I would like to present my paper, Beta-3 Adrenoreceptor, Boon or Bane in Heart Failure. So for our objectives of the study, we are here to study the role of beta-3 adrenergic receptor up regulation or down regulation in heart failure. Our specific objectives is to discuss the effect of beta-3 receptor agonism and antagonism, and to review the effects of 4 selected beta blockers on beta-3 receptors. And we've used our references in PubMed, Herdian, and Cochrane. So what we've used is that we've searched using beta-3 adrenergic receptors, beta blockers, heart failure, and selected beta blockers such as metoprolol, bisoprolol, carvedilol, and nebivolol. As you can see in the table shown in your right, these drugs are shown to be efficacious in reducing mortality in human studies on heart failure. That's why we've selected them for our discussion today. So for a brief background on what are beta receptors, so what we know on beta receptors, they are divided into 3 main subtypes. So your beta 1, your beta 2, and your beta 3. So your beta 1 is mainly found in your heart, so it increases your heart rate and your contractility and automaticity in different parts of your heart. And your beta 2 are mostly focused on your vascular system, and they also affects smooth muscle relaxation in your bronchi. So your beta 1 and your beta 2 receptors are found in your heart, causing positive inotropic and chronotropic effects on the myocardium induced by endogenous catecholamines. So as I've said earlier, beta 1 receptors are more predominant in the heart and responds better with norepinephrine than your what we call beta 2 receptors. So what are your beta 3 adrenergic receptors? So in 1989, they have shown to have pleiotropic effects on number 1, brown fat thermogenesis or lipolysis, which is highly expressed in your adipose tissue. Number 2, they have effects on your ileal relaxation. And number 3, they have effects on solute muscle glycogen synthesis. However, as the years gone by, they have tried to explore the possible effects of beta-3 adrenergic receptors in your heart. So in this figure, this is a graphical representation of the molecular level of your different receptors, your beta 1, your beta 2, and your beta 3. So in your beta 1 and your beta 2, as you can see the encircled figures in the screen. So beta-3 adrenoreceptors lacks the particular amino acid sequences targeted by your protein kinase A or your PKA and G protein coupled receptor kinase or your GRK2, making it resistant to catecholamine-induced downregulation in chronic heart failure patients. So in chronic heart failure, as we know, beta-1 adrenoreceptors and beta-2 receptors are desensitized and downregulated because of chronic high plasma catecholamine levels. So, compare it to your beta-3 receptors, it is coupled to two different isoforms of G proteins, your G-alpha stimulatory subunit, which your beta-1 has, and your G-alpha inhibitory subunit, which the beta-2 receptor has. So, when we try to activate the beta-3 adrenoreceptors, the downstream pathways would lead to activation of your nitric oxide synthase pathway, which we will discuss later on. So in this figure, it is said by DeLucia 2018 that there is approximately a 4:1 ratio of beta-1 receptor to beta-2 receptors in the heart. So it's around 80% for your beta-1 and for your beta-2, they have around 20% representation in your heart with an insignificant amount of beta-3 receptors in the normal heart. Usually, they are detected in only around 5% of myocytes, but are abundant in non-myocyte cells. So what happens to your beta receptors in heart failure? So in patients with congestive heart failure, beta 1 more than beta 2 adrenergic receptors are downregulated. This is due to the phosphorylation by camp-dependent protein kinase or beta adrenoreceptor kinase, which would lead to a decreased expression in cardiac myocytes. However, it is shown by the researches that we've reviewed that beta-3 adrenoreceptors are upregulated. So as we discussed before, they don't have the same pathways as the beta-1 and the beta-2 receptors. They lack the phosphorylation sites for camp-dependent kinases. So there. Okay, there. So in this graph, so in the graph, the letter B, so it is said that the ratio becomes almost equal in a failing heart when beta-1 receptors are downregulated, correlating with the severity of your heart disease. So on the other hand, your beta-3 receptors increases in in quantity. So they are overexpressed in heart failure and hypertension, and then they increase more when conventional beta blocker therapy according to Newton. So what happens is that when these beta-3 receptors are upregulated, the research says that they counteract downregulation by the signaling of your adenylyl cyclase or your G-alpha inhibitory subunits. So it makes the heart more sensitive to catecholamine stimulation, hereby increasing your inotropy. It tries to blocks the effect. It tries to block the effect of your catecholamines and prevents further beta-1 and beta-2 adrenoreceptor downregulation. So what happens? So when we try to activate or stimulate your beta-3 adrenergic receptors, your downstream pathway would lead to the activation of the nitric oxide synthase pathway, and that it suppresses the NADPH oxidase. So what happens briefly is that the activation of your NOS by the upregulation of your beta-3 would lead to the production of your nitric oxides, which we would discuss later on, and that it suppresses the NADPH oxidase, bringing about its antioxidant effects that downregulate your ROS. So what we see usually is that the activation of your beta receptors would increase your CAMP levels. They increase your cardiac contractility. So in theory, what the research says is that when you activate your beta-3 receptor, it decreases your CAMP levels. And the correlation is when you have a decrease in your CAMP levels, you have a decrease in your cardiac contractility. So it is in contrast with the activation of your beta-1 and beta-2 receptors, and it is said that it's because of the nitric oxide. So what is the nitric oxide that I'm talking about? So it's associated with beta-3 adrenoreceptor activation. And what it does is that it expresses a negative inotropic effect because of its vasodilation properties. So in the normal heart, they say, the researchers say, that it can stimulation of your beta-3 inhibits your cardiac contraction and relaxation. But in heart failure, the negative inotropic effect is actually beneficial because it prevents your pressure overload. It acts as an endogenous beta-1 adrenoreceptor blocker. So they actually help or augment the down regulation coming from chronic catecholamine release in patients with chronic heart failure. So here is a figure that we've made to easier discuss the effects of the stimulation of your beta-3 adrenergic stimulation. So what it does, it activates your nitric oxide pathway. So it promotes two main things. So the vascular effects and then the myocardial effects. So what it does, we try to increase or try to activate your cyclic guanosine monophosphate or CGMP and protein kinase G or your PKG. So your vascular effects would lead to an increase in your endothelial function that would lead to a vasodilation and improvement of your coronary perfusion. For your myocardial effects, so your PKG activation, your NOS pathway activation, it can increase your myocardial relaxation, preventing excessive oxygen demand and ventricular remodeling. Also, your PKG activation reduces your calcium oscillations that prevents arrhythmia, hypercontracture, and sarcolemmal rupture with lead to cell death. Also, we've discussed that it suppresses your NADPH oxidase, bringing about its antioxidant effect. So what it does, it downregulates the reactive oxygen species or your ROS, which deactivate your NAK ATPase pump. So you when your reactive oxygen species is present, it can cause your endothelial inflammation and myocardial fibrosis. So when we try to activate your pump, it decreases your intracellular NA, so it restores your pump activity, which would improve your contractility. And then all these effects would theoretically lead to a hemodynamic improvement in patients with heart failure. So there are a lot of studies proving the effect of nitric oxide synthase of nitric oxide in heart failure, such as the research has shown there. So for the next chapter of our discussion, we selected a few beta blockers in our study. So the first one that we would like to discuss is nebivolol. So nebivolol has greater selectivity of beta-1 receptor blockages than other beta blockers. In the graph shown here, it is shown that nebivolol has a greater blockade of your beta-1 compared to your bisoprolol or your metoprolol. So it is said by Cohen as Arazi that nebivolol has noted to have beta-3 adrenoreceptor agnostic function, and it tries to exert its vasodilatory effect to the production of nitric oxide derived from the endothelium. So what it does, because endothelial dysfunction caused by oxidative stress is a mechanism involved in elevated BP, what it does in the studies, nebivolol showed reduction of oxidative stress. So it contributes. So there are many studies that try to discuss regarding the benefits of using nebivolol in patients with heart failure, but we're going to focus on the study on Arazi. So they propose a synergistic action of nebivolol in the treatment of heart failure. They subdivided nebivolol into two isomers when they are ingested in our body, the L isoform and the D isoform. So the D isoform is the main mechanistic function because it acts as your beta-1 blockade. So what it does, it reduces your heart rate at rest and on exertion, and it also reduces your systolic and diastolic BPs without causing adverse effect related to your beta receptor 2 blockade and maintaining the balance with the alpha receptors at your vascular level. In comparison with the L isoform, so what it does, you have your nitric oxide release in your endothelium, which causes vasodilation, which leads to a reduction of peripheral vascular resistance. There is improvement of remodeling and of arterial stiffness, and in the increase of systolic volume and ejection fraction, maintaining the minute volume. So for the next drug of interest, so we're going to discuss about metoprolol. So metoprolol is known as a cardioselective inhibitor of beta-1 adrenoreceptors with minimal to no effects on beta-2. It is shown in studies that it can upregulate beta-3 adrenoreceptor expression. So there are animal studies such as the study from Sharma and the study from Zhao that there is upregulation in your beta-3 adrenoreceptors, but it leaves lot to be desired for human subjects to be tested if there is effect on beta-3 agonism in using metoprolol. For your bisoprolol, our next drug of interest, so it is a highly selective blocker of the beta-1 adrenoreceptors. It is said that there is no effect on nitric oxide generation. However, it attempts to promote its vasodilation by its beta-1 effect. In studies that we've seen, for instance, in pastel de Groot or in Schnabel, they've shown that the bisoprolol has a neutral effect to beta-3 adrenoreceptors. And for our last drug of interest in carvedilol, we know that carvedilol is a non-selective adrenergic receptor blocker. It is also an alpha-1 adrenergic receptor antagonist, and it exerts its peripheral vasodilating effects using the alpha-1 receptor. So when it does its vasodilating effects, it attempts to decrease its total peripheral resistance. In studies from Moser, it is shown that anti-proliferative effects on smooth muscle cells, and it also has effects on inhibiting the oxygen-free radicals. In studies that we've seen in Canavo and Co., it is shown that carvedilol is known to downregulate beta-3 adrenoreceptors alongside your beta-1 and beta-2 adrenoreceptor expression. In limited studies using monkey cells, coming from studies from SNAB, carvedilol binds to your beta-3 adrenoreceptors. So with the interest of the drugs that we've discussed, this is the summary of what we're trying to show you. So in metoprolol and nebivolol, they show to have upregulation of the beta-3 adrenergic receptor. And yes, in the table shown here, there are studies that show that they do generate nitric oxide. Your bisoprolol has neutral effects on your beta-3 adrenoreceptors, and your carvedilol has shown to have downregulatory effects in your beta-3 adrenergic receptor action. So for the conclusion of our review, we've determined that beta-3 receptor stimulation would lead to the activation of the NOS pathway and suppression of your NADPH oxidase that causes vascular and myocardial benefits, leading to hemodynamic improvements in patients with heart failure. It is also said that the use of beta blockers, regardless of mechanism and receptor activation or blockade, ameliorates symptoms in a failing heart. However, the function of metoprolol, nebivolol, bisoprolol, and carvedilol on beta-3 adrenoreceptors are varied, and of course, further studies are recommended and are encouraged so that it can shine a light in order for us to better understand the mechanisms of beta-3 receptors. And studies on nebivolol and metoprolol suggest the upregulation of beta-3 adrenoreceptors to exert in nitric oxide production. With that, I thank you for listening to my presentation.